Good morning, Akshay. Dr.
Yes,
Akshay.
yes, Dr. Srinivasan, good morning.
Good morning. Great. So we'll wait for the people to join.
Sure.
Yeah. Meanwhile, I'll just display the instructions to participants.
Okay. And later on, maybe I'll say about 40-45 minutes is my lecture, and then later on it would be it would be more say useful if we have a kind of interactive question answer session. So which Dr. Srinivasan also mentioned that the questions will be posted in the chat box, and I will try my best to answer whatever I can. And so let's take it forward. I'll just share my screen and I'll begin.
Yeah.
I guess my screen is visible.
Yes, Dr. Akshay. It's visible.
Okay. So I have kind of uh, divided my talk into the following 6 broad headings. Just a very brief introduction, scope of regulatory toxicology, and then the purpose, why, how, and when, and why these regulations came into being. A little bit about the risk assessment and hazard identification, then the various regulatory agencies. I would just be mentioning the regulatory agencies, areas where they have their role to play, and then um, briefly about the various uh, new drug applications, investigation new drug application. So coming to the this genesis of regulatory toxicology, why and how there was a need a need was felt for regulatory toxicology to evolve as a separate discipline. Pharmacology, toxicology, various studies, experiments, etc., were going on since a very long time, have been going on. But then there was this need that was felt to have some sort of a regulation, a control have certain rules under which various studies are performed, or certain rules under which it is essential to get certain studies done, to perform certain studies, experiments before you allow the product. So when I say product, it can be a drug, it can be a nutraceutical, a food additive, feed additive, or things which are used, say, for packaging. So for anything new that is being introduced, there are certain uh, guidelines, rules which need uh, to be fulfilled, to be followed, and this has evolved over time. Not all of these regulations came in right at the very beginning. As and when a new need, a new necessity was felt, a new regulation was brought in. Uh, Very briefly, to go about, about the history in toxicology. Initially, as I'm sure all of you are aware, it was mainly to do with uh, the focus on poisons. What are the various poisons or the toxins? And so, initial days, it was mainly kind of focusing only on the existing toxins and poisons. Using that knowledge, slowly people then started developing antidotes, and based on, then once these antidotes are developed, and then uh, the use of the poisons, more was learnt about their use and their effects. Then they started getting in certain rules to say, to prevent the illegal use or to prevent the misuse of toxins and poisons. Over a period of time, then it slowly, then they started extending the same to use of drugs, dispensing, how a drug should be say dispensed, how it should be prescribed, how it should be dosed. These say specifications came in at a later stage. uh, this is something which all of us know, that it is the dose which decides whether something is a toxin or is toxic or no, or whether it is poisonous or no. And here again, when I say it is a dose, like when we talk about either preclinical toxicology studies or uh, any other say safety evaluation, safety pharmacology studies, uh, a lot of input is required while we decide the dose, because the dose that we need to use in a particular study would decide the fate of that particular chemical. So unless we know, or unless we have certain data, some information about the efficacy dose of if I take the example of a new drug of a pharmaceutical, initially we would have some information about the efficacy, the dose at which it is efficacious, and then there are certain set say formula wherein we can like extrapolate the doses from uh, a rodent study to a human study, or from an in vitro to an in vivo. So taking all these into consideration, the dose is calculated or decided when we plan a toxicology study. Again, so initially, as I just mentioned, it was mainly uh, after poisons, drugs, chemicals. Then they slowly expanded the scope, and then they started including the studies on the effects of xenobiotics, and also other chemicals. Any chemical which might be used either as a drug, or which might be used in any other process, uh, say in any other manufacturing process. But then, if there is if there is a chance of human exposure, and how much of that exposure um can be allowed, how much of that exposure is safe, and what is the level above which it may cause some harmful effects to the human who gets exposed to that chemical. That also then slowly started getting into the ambit of toxicology. Over a period of time, as medical practice itself evolved, medical practice, say there were developments when there were medical devices which were being used, and before any such device was allowed to be used in for human applications, their use also began to start getting regulated under certain broad guidelines. And so that's how started with poisons, drugs, chemicals, biomedical devices, and then slowly it also went on into the realm of environmental safety, environmental or rather ecological safety. So now when we talk about regulatory areas, regulatory guidelines, there are also there are guidelines which decide and uh, which have a control over what are the kind of chemicals that can be used, what are the levels that can be say released into the environment, and all such areas. So in order to be able to say govern, or in order to decide the rule or the regulation that has to be applied, toxicology, say certain area, certain uh, the branches in toxicology are being used. So to start with, initially we have the mechanistic toxicology. So mechanistic toxicology is that branch or that area which deals with how exactly the change is being brought about in the biological system. So when we use a chemical or a drug, we see some change in the biological system. So how it happens, why it happens. So basically, it actually describes the cellular changes, the biochemical changes, or the molecular changes. And the early days, it was to do mostly only in a living system, in a living organism. But as I said, over a period of time, this then also started extending into the environment and other ecological features. After mechanistic toxicology comes actually descriptive toxicology. So once uh, the mechanism of action has been identified, or uh, elucidated, that it has to be described in a manner based on which we are able to draw some conclusions. So we know that a chemical or a drug is causing some change. For example, it is causing a change uh, in the metabolic functioning, in the liver functioning, or in the kidney functioning. We need to know what exactly is this change. We need to describe what exactly is the change, and only then we would be able to use that information, that data, to set up or set down the rules under which we can allow or not allow the particular chemical which is being tested. And so uh, based on the descriptive toxicology input, we can then plan our study, or we can then design a particular study to get the particular say answers to our questions. If it is a new chemical, again I keep taking the example of a drug or a new drug. If we know that it is being say developed for example, say for a diabetes uh, for condition of diabetes, we would expect certain other side effects also along with it, as we get the input from the other existing anti-diabetics. So again, using those descriptive toxicology endpoints, we will design the study for our existing new drug. So once we have all these inputs uh, of mechanistic and descriptive, we come to regulatory toxicology, that which is the main say the crux of the talk today. And so the major the main end point that we are looking at in a regulatory toxicology study is that we need to decide, or at the end of the study, we need to be able to decide whether a drug or a chemical poses a risk when it is used in humans. First is whether the risk is there or no. Secondly, how high or how low is the risk. And if the risk is sufficiently high, then there are no doubts that will be dropped. That will be, further development of that particular chemical or drug would not be taken up. But however, if there is a very low risk, and the benefit out of the use of that is higher, then that is where that is where the decision has to be taken with respect to the risk benefit ratio. I'm sure you all would have heard of this term. The risk benefit ratio is something which keeps coming up again and again, again and again, whenever there is some new guideline to be formed, a new say regulation to be implemented, or whenever a new chemical or a new say substance material is being developed uh, for human use. These inputs are also then used for product registrations. And in case if I take the example of say nutraceuticals or certain food additives, feed additives, all of you would know that there is a lot of fortification of various say micro minerals, micro elements, which is being done in various foods, milk, atta, etc. So all of these have certain limits up till which it is allowed, beyond which it is not allowed. So how this limit is decided is based on what is the RDI for that particular uh, mineral, micro mineral. Having said that there are certain regulations, rules, etc., we need to be very careful and critical in deciding what is the amount of uh, data that is required, what is the amount of regulation that should be implemented. Now, if we have too much of uh, say too much of data that is generated, too many rules that are in place, in actually the entire process of going through that would only delay the uh, say the further development of the molecule. So what we need to be careful about is that the data or the information that is generated, the data that is generated should be sufficient. It should not be too much, which will actually bog down the entire process. It should be sufficient to be able to address any technical or legal issue which might arise during the process of development, or it should be sufficient to be able to take this decision of the risk and benefit, whether uh, the end usage or the end outcome of using that is more beneficial. Though there is a certain amount of risk involved, the benefits are higher. And all the data that is generated, or the amount of data that is generated, should be also of high quality. Having actually there needs to be a balance between the quantity and quality. We should have sufficient amount of data that will provide the evidence based on which a particular decision is taken, uh, in terms of its usage, in terms of legal issues, in terms of technicalities. The any data that is generated under the realm of regulated talk should be able to provide evidence that is required to help the decision that is being taken. Now, as I had said initially also, risk benefit ratio is what is key whenever we look at uh, say the development of a drug or a chemical. As was famously said by Dr. Dole, that toxicology is what we do, but why we do it is what is the outcome, which eventually leads to the outcome of risk assessment. We do a lot of toxicology studies. Maybe initially it is for say in initial drug development process, but then the ultimate outcome or the ultimate goal that we are doing it is to be able to decide the to be able to decide or to be able to estimate the risk that might be involved. So, coming to risk assessment, it is basically a quantitative assessment of the potential effect on the human health. If you go by the definition of it, but then the broader picture, it includes the environmental significance of various types of exposures. For example, when we use say pesticides per se are toxic. They are meant to be toxic so that they are able to say destroy the pests that affect our crops. But then when we talk of risk assessment here, we are referring to the, or we are looking at the person or the people who are involved in the usage of those pesticides, or the usage of those agrochemicals. So certain amount of regulations are in place with respect to the use of an agrochemical. agrochemical usage is required for controlling and for treating pests, etc. But then the usage needs to be regulated. And the other aspect in when we are doing a risk assessment study is we cannot go with just the acute study data, acute lethality, because acute lethality may not always be a reliable measure of the long-term hazard. For example, a chemical which has very low acute lethality cannot just be designated as a safe chemical, because it might have long-term effects, or other like chronic effects. It might cause it might lead to carcinogenic changes and other or maybe genotoxicity. So when we talk about risk assessment, it is not just a single study or a single type of study uh, which is used for data generation and for uh, for drawing a conclusion. But then there are acute studies, then there are chronic toxic, there are long-term studies. There is a certain amount of mathematical modeling which also goes in. Uh, there's one particular point which comes up when in a risk assessment study is with respect to the uncertainty characterization. So uncertainty characterization, basically we use in vivo tools and also in silico tools. Both are used based on the data that we generate by the conduct of an in vitro or an in vivo study, and the databases that are available using various say softwares or in silico tools. Both are put together, and then there is a conclusion drawn about the uncertainty characterization, based on which we then take a decision with respect to the risk assessment. Uh, Very broadly, just to put the various steps of risk assessment in a pictorial form or in a graphical representation, the first main goal is to identify the hazard. Uh, we know that there is some that some like or we would go with the assumption that there is some likely hazard, probable hazard that might occur. So we need to first identify that hazard. Once we identify the hazard, we need to determine at what level of exposure do we see that particular outcome. Now, if we know that there is a hazard that is likely, but then when we use that particular chemical, we are not reaching that level of exposure which causes the hazard, then obviously the risk is very low. And but however, if in case the usage of the particular chemical itself is such that there is a likelihood of high exposures, and if there is a high exposure, then obviously the hazard will get manifested. And this again, when we do an exposure assessment, this exposure assessment is goes hand in hand with the dose response. Low doses, no hazard. Higher doses, increasing hazard. High and still higher doses might eventually, cause say even deaths. So the dose response assessment and the exposure assessment, they both go hand in hand. And once we are able to get inputs on all these three, that is, we identify the hazard, the exposure assessment, and the dose response, then we do the risk characterization. So what is the extra risk of the health, or what is the extra risk that the particular population that might get exposed to that particular hazard? Based on this, then a decision is taken within the existing, say, regulations, guidelines, as to whether to allow or not allow uh, the particular, say, chemical. Uh, Mostly, whenever as I'm talking, I'm taking the example either a drug or a new drug or a chemical in most cases. But then this would vary slightly depending on what is the particular um, item that is being tested. For example, if you're looking at a chemical which is used as a packaging material, say for a very long time we used to use the bisphenols were being used in plastics. So uh, once they realized that bisphenol A and other analogs are toxic, they started developing other alternative forms which are now being used. So the exposure assessment that we do for a drug or chemical and the exposure assessment we do for an ingredient of a packaging material would be different, but the end goal is the same. That is, what is the level of exposure in the human population and what is the effect or the harm that might be caused uh, at that level of exposure. The outcome or the end goal is the same, but then the extent of analysis, extent of assessment would vary. Just a few slides on what are the different types of testings that are being included under uh, the regulatory toxicology. Initially, the first is the in vitro test. In vitro, as the word suggests, is in the glass. So it is something which is done outside a living system, and it involves isolated tissues, organs, cells, cell lines, primary cell cultures, etc. And it is useful in initial stages, initial detection, initial identification of biochemical or genetic effects that might be caused by the use of a particular uh, chemical. Uh, it has certain advantages and disadvantages. The biggest advantage being that in the present time of where there is a thrust for the reduction in the use of animal testing, in vitro test methods help. We are able to use it as a high throughput screening tool, and only those chemicals or those drugs which are really which show some potential are being taken into in vivo animal studies further. But then certain disadvantages in an in vitro system, we are not able to evaluate the biological effects of a whole body exposure. It is or the exposure in an in vitro system is very limited. The kind of data that can be generated is also a little limited compared to an in vivo test. In vivo, as the name, as the word means, that it is in a living system, and so this is done in various living systems, either say in rodents or in the canine model or in the non-human the models. Then you have various ecotoxicology models like zebrafish, algae, daphnia, etc. And a lot of these studies, a lot of in vivo testing goes into especially goes into the preclinical safety evaluation for drugs, chemicals, for cosmetics, animal testing to some extent for certain types of say cosmetics are now is now banned, but then for drugs and chemicals, a lot of in vivo testing still being done. The advantage is that we are, as the main drawback of the in vitro system, that is we are not able to evaluate the entire biological system. That is overcome in this, wherein uh, when we administer the chemical to a rodent model, in a rodent model, the overall effects of it are can be evaluated. There are different types of studies which are included: acute, subchronic, chronic. Then you have multi-generation studies. You also have reproductive toxicology and carcinogenicity studies. So um, by the entire battery of tests, uh, we are able to evaluate the particular chemical and the drug or the drug uh, in its entirety. That is, if it is, as I take the example of a drug being developed for diabetes, but then if it is going to have some effects on the cardiovascular system or some effects on say the respiratory system, these outcomes can be evaluated in a in vivo system. It is not always possible to do so in the in vitro system. Certain points which apply to both in vivo in vitro systems, they are basically used to predict the inherent say hazard that might be caused by the chemical property, by the chemical nature of the drug that is being developed. Whenever a new drug or a new say chemical is being developed, uh, before going into in vivo in vitro studies, we would have data on its physicochemical properties, on the class of compound that it belongs to, class of compounds that it belongs to. So based on those inputs, certain conclusions are drawn with respect to the probable say metabolism routes, metabolism processes, probable effects that it might cause in an in vivo system. So these can be evaluated or rather screened in both in vivo and in vitro studies. And one point of caution that, of course, you all are already aware, in vitro studies or outcomes of in vitro studies cannot be directly extrapolated or cannot be directly used to predict the response in an in vivo system. In vivo, in vitro studies should always be utilized complementary to one another, not as a replacement for one another. You need to conduct both in vivo and in vitro studies, and but then what is the in vitro study that is required, what is the in vivo study that is required, that would be governed by the particular regulatory guideline that you need to fulfill. So all your studies which are being planned, which are also designed in a way to meet or to fulfill a particular say a particular guideline or a regulatory submission when you are making. So what are the inputs that that particular regulator needs before say evaluating the chemical and allowing or not allowing the chemical? The other important point is that there should be a regulatory acceptance for the particular study that you are that is being planned. Not all countries accept all in vitro data. There are several in vitro studies, though, which are being performed at the R&D level for several years, or that are being performed to generate data, may not always have a regulatory acceptance. So we need to be sure that the particular study that has been selected, that has been designed, uh, in tune with the regulatory requirement. To take the example of OECD, there are several in vitro OECD studies, or rather several in vitro studies which have the OECD acceptance. And if you look at the OECD guidelines, there you have in vitro studies for skin sensitive. You have in vivo and in vitro studies both for say for skin sensitization, skin irritation, for corneal opacity studies, for mucous membrane irritation studies. So there are several in vitro studies which have been granted regulatory acceptance, but there is still, so any new study that is being developed would need to undergo this entire process of uh, intra-lab validation before it can uh, seek say regulatory acceptance. The other class of studies that are being performed now, this is actually a recent development last few years, is the in silico toxicology. In silico methods basically uses the computational approach and uses data that has already been generated for uh, existing chemicals, existing class of chemicals. The biggest advantage is that, as is the advantage in any other computer aided system, is the speed at which you can process information. So a very large set of molecules, compounds can be screened very rapidly using in silico methods, and uh, a broad analysis can be made about the probable toxicity of the particular chemical. But however, point of caution here again is that this is basic. This is mostly based on data that is available in the public domain, based on data that is already has already been generated, and based on say the structure of the new chemical or compound, a certain amount of conclusion is drawn with respect to the probable toxicities. This is the advantage. One advantage which I said initially is the speed, and secondly, it also helps in meeting requirements of the 3R principles. That is replacement, reduction, refinement to reduce use of animals. We can use this as a high throughput method again to screen several compounds, and only those compounds which um, show certain promise with respect to the structure activity, or with respect to the safety, with respect to the low hazard levels. Only those compounds can then be taken further to the in vitro in vivo testing. This helps in a lot of saving of time, a lot of saving this of money, and therefore it is used as a quick screening method. But then this again, what I had said a few slides earlier, that we need to always keep in mind that this is uh, the conclusions are drawn based on existing data. So we would like to, like how initially the in vitro data cannot be directly used to predict in vivo outcomes. Similarly, you can use the in silico methods only as a screening or as an initial tool uh, to decide what kind of in vivo study or in vitro study you need to do. You cannot completely use this to replace in vivo in vitro studies. So this is just to give an outline of how the in silico toxicology is um, interlinked with with various in silico methods, like you have data generation, you have the you have the structure activity relationship, and then one particular point which is used very often is the method of read across. So based on toxicology data of existing chemicals of a similar class, we read across and we are able to predict the probable toxicity outcomes of a new chemical. Uh, most of these decisions are based on the QSAR relationship, or the activity of the particular chemical, and that is what forms the basis of the in silico test methods that are being employed. Uh, so, having spoken about the different types of uh, the tests that are done, a few slides on the different regulatory agencies uh, uh, which have a role to play in safety evaluation of various drugs, chemicals, food products, etc. The Food and Drug Administration, they lay down the broad guidelines for testing of impure and of also drug substances. Environmental Protection Agency, they are involved with the environmental outcomes, ecological safety. Then the Occupational Safety Health Administration, with respect to the outcomes, or with respect to the exposure levels for the at say in a manufacturing process, or in an assembly line process. The others are the ISO, OECD, and the European Medicines Agencies. All of these agencies are interrelated in the sense that the end goal is with respect to safety evaluation is similar. Uh, the class of compounds, or the class of drugs, or the class of materials that they apply to may vary a little bit, and OECD principles, or other OECD studies, are the studies which are very broadly, which have very broad worldwide acceptance. Most of the GLP studies um, uh, are being governed by the OECD guidelines, OECD principles. How, of course, specific countries have their own the regulatory requirements in addition to the OECD, and this also varies with certain say geographical regions. ICH has its own International Committee on Harmonization. They have their own say regulatory guideline, regulatory requirements to be met, to be fulfilled. Then recently, 2019, in India, we had there was a total revision and reintroduction of, not reintroduction, there was a revision of the Schedule Y. So it is now called New Drug and Clinical Trial Rules 2019, which in which there are certain changes that have been made with respect to the type of studies, with respect to the animal group size with respect to uh, the species in which a particular study needs to be conducted. So these are certain changes which have been brought in into the new drug chemical trial rules in India. FSSAI, ICH I already spoke about. FSSAI is mostly with respect to the food ingredients, food additives, food packaging. All these are broadly covered by the FSSAI guidelines. And in recent times, FSSAI is also now involved in the use or in the methods of preparation of even the street food. You would have seen or read about this in the last couple of years. There are certain guidelines which have been formulated and which are being implemented even for street food vendors. Then safety pharmacology studies mostly are covered under the ICH S7A and S7B. New drug clinical trials I already spoke about, and similar to the GLP, good laboratory practice, you also have a whole set of good clinical practice guidelines which govern the uh, various phases of clinical trials that are being conducted. Central Insecticide Board is mostly concerned with the various agrochemicals, pesticides which are being used. Very recently, there has been a set of guidelines for the biopesticides. Most of the guidelines rules were initially for the agrochemicals, and now they are also being developed for the biopesticides. It is still in initial stages. Once they are finalized, these will be notified. Just a couple of slides on this might be a kind of repetition to most of you, because in the area of say pharmacology, toxicology, investigational new drug application for any new chemical or a new drug, it's basically the organization or the company is asking for an authorization for clinical investigation. This is being done. This has to be taken. The investigation new drug application has to be made, and say permission taken, even for transportation and distribution across different areas. So when you need to test a particular chemical, it needs to it needs to be tested in more than one geographical area, more than one uh, say ethnicity. So, and it involves certain amount of transportation. So this is what is permission for these applications are what is applied for under the investigational new drug application. It should include information on the quality of the chemical, on of the chemical nature of the structure activity relationship, then all your non-clinical safety data, which, as I mentioned, the in vivo in vitro studies, and what is the proposed say clinical protocol that is being suggested. New drug application is made for seeking marketing approval. Here we need more detailed information, like with respect to the pharmacokinetic, pharmacodynamic properties of the particular chemical, uh, the initial clinical studies, and then characterization of impurities. All this data is also generated before you make a new drug application. And what is the information that needs to be mentioned on the label, on the packaging insert of the particular chemical, is also mentioned in this application. And once approval is granted for all these aspects, only then it is allowed to be taken into the market. It is allowed to be taken into the next stage of the trial. And finally, abbreviated new drug application is uh, a drug or a chemical that is being used in a particular say disease condition, or it is being used as a branded drug, and now a company is applying for the generic form. So this is what is covered under the abbreviated new drug application. Here, the most important, apart from, see, since it is already an existing drug, the toxicology and other data would be already available. The most important is that the applicant should be able to prove scientifically that uh, this particular generic form is in all ways equivalent to the existing branded formulation. So basically, the bioequivalent studies and the other bridging studies, those data is what is important in an ANDA. And since it is a generic form of an existing branded drug, other preclinical data which is already available may not be included in the application. Since that is already available in the public domain, any review of that data is possible. The most important point here is the bioequivalence to the existing drug. So that is about what I wanted to talk to you all about today. So basically, initially, just a very what we had was a brief say introduction scope in regulatory toxicology. What are the different types of studies that are being done to generate data for the regulatory requirements, the various regulatory agencies that play a role, and very briefly the new drug applications. I'll stop here and maybe then take the questions. I'll just stop sharing the video and then we can continue.
Uh, Thank you, Dr. Akshay. It's a very good presentation and a brief overview on the regulatory toxicology. So we have only few questions here. That recently there is one question by. I'll just read out.
Sure.
Which guidelines are advised to be followed for preclinical toxicity evaluation of traditional medicine system formulation in India, OECD or Schedule Y or any other?
So it depends on what is the if it is being developed, if that particular say traditional formulation is whether it's being developed as for a therapeutic use or as a nutraceutical or as a general wellness product. So if it is being developed for a therapeutic use and for a regulatory submission within India, you would need to meet the new drug clinical trial rules. But then if it is being developed not as a drug or not as a therapy, it is being developed as a wellness product or just as a additive, food or a feed additive, it would not need to undergo the particular preclinical testing like is being done for a drug. So there, based on the data that is available in your Ayurvedic texts, in your traditional medicine texts, that data would need to be, or say that information would need to be submitted for approval for marketing, for usage.
Okay. Yeah.
Yeah.
Just a second. So there is not much questions today, and I have few questions from my side.
Please.
Is regarding the veterinary toxicology, veterinary products. So we don't have a kind of a guideline available for safety evaluation of veterinary drug products, right, in India. Yeah. So how is it like if a company is coming forward to go for launching a new drug, veterinary drugs? So what guidelines to be followed for the veterinary drugs?
So this is something which I would also need to look at. I would also need to get some information because even as you said, veterinary products. I mean, if it is a new drug or a new chemical being developed for veterinary usage, right now, what are the guidelines within the country? I am not sure myself. I would need to look that up. But then, if you look at the global scenario, global picture is mostly say efficacy studies need to be done, and then once we are able to show the efficacy of the particular new chemical that is being developed, if it is a new chemical, if it is some alternative to, or if it is different analog of an existing chemical, then maybe we would need to show the bioequivalence or the equivalence with respect to efficacy. Uh, these are the two main things which come to mind right now. But then, as I said, no specific guideline or regulation that I am aware of. I would need to check on this.
Okay, there is a similar question by Dr. Mukesh Subhas Patil on which guideline are to be followed for establishment of toxicological investigation lab in veterinary field. Maybe he's asking about the guideline to be followed for the establishment of toxicological investigation. Maybe he's talking about GLP, but in veterinary field he's asking.
So GLP is basically, I mean, it's basically say a quality system. So it does not differentiate between whether you're doing it for a human drug chemical development or a veterinary drug chemical development. GLP basically deals with systems to be put in place for conduct of a study. Say, for example, when you are conducting a particular study, it should be planned in a particular manner, it should be conducted in a particular manner, it should be reported in a particular manner. You should have SOPs for each of these three phases. You should have SOPs for each of the activity that will be conducted in any of these phases. For example, a simple activity like weighing an animal, there should be an SOP for weighing the animal, there should be an SOP for the usage of the balance. So GLP guidelines, excuse me, those are very clearly available on our on the NGCMA website, National GLP Compliance Monitoring Authority, the website. So, in order to set up a GLP certified lab, you would need to fulfill those requirements, which are as mentioned on OECD sites, NGCMA sites, etc.
Okay. Next question is on Unani medicine. And before that, Dr. Nitin Bhatia from INFA probably he has just given a statement that there is an outlined tox study requirement for animal health product. There is an outlined tox study requirement for animal health product.
Okay.
Maybe it's not where it is available. If Dr. Nitin Bhatia can give more information, that would be useful. And for next question is for testing Unani formulation in the form of toothpaste. Do we need to just a minute? Do we need to do a regular toxicity testing? What kind of data we need to produce for marketing purposes? Maybe he's asking for toothpaste. Unani medicine in toothpaste.
So again, Unani medicine, if it is something developed out of Unani medicine, then it would come under the ambit of the traditional medicine systems. So requirements for traditional medicine systems would need to be fulfilled. But having said that, like this particular case, since he has asked for it to be used as a toothpaste, so toothpaste usage, toothpaste exposure levels are very different. So it would not be as for a drug or for a chemical, it would be for a consumer product. So that there in that case there would be ISO guidelines which you would need to look at and accordingly plan the study.
Okay. Yeah, Dr. Nitin Bhatia has now explained toxicity studies for veterinary products is already outlined with the CDSCO website. I would maybe probably I can unmute Dr. Nitin Bhatia if we can explain a little more on that because this is a veterinary forum. Probably your information will be useful to most of the veterinarians in the group. Dr. Nitin Bhatia.
Yes, sir. I think most of you know me. I'm Dr. Nitin Bhatia. I'm working with Intas Pharma. I'm a MBSc in medicine and with a minor of pharmacology. I have been very regular with ISVPT. So if I saw the question coming up and I thought that it will be better for me to respond on that. The government has already outlined the rules and regulations for veterinary products. They are with the same ministry as Ministry of Health for Human Healthcare. The products are very much outlined very similarly to human healthcare. And in terms of tox studies, we have to do studies on mice and rat. It is clearly there on CEDESCO website, which is the Ministry of Health website, and it is fully outlined for animal healthcare products. In case
Oh,
any more doubts are there, I am there available to answer that.
okay.
Right, Dr. Nathan. Thank you for the input, Dr. Nathan.
Welcome.
Thank you, Dr. Nitin. The next question is about does generic medicine require preclinical toxicology testing?
No, as I said I think towards the end, the generic drugs, when you apply for an ANDA using a generic drug, mostly you need to show the equivalence to an existing drug that you are trying to um, equate against. So preclinical data would already be available. You would need to say assimilate that data, but you need not conduct a preclinical study again.
Okay, so the same person, I think Dr. Pramod Bharani is asked, does veterinary homeopathy drugs require any trials before using in the field? Again, as you already explained, it comes under traditional medicine. So does veterinary homeopathy drugs require any trials before using in the field?
So I think it would be a good idea to also look at this particular input which Dr. Nitin just gave us with respect to veterinary drugs. If you consider it as irrespective of whether it's a homeopathic or an allopathic, it's a veterinary drug. It needs to meet the particular requirement. Then accordingly, that particular study would be required. But maybe the CDSCO link which Dr. Nitin was referring to would be a good place to look at that.
Okay. There's one more question from Dr. Ratandeep. Which regulatory agency in India is responsible to regulate MRL, that is maximum residue limit of veterinary drugs in animal products? MRL.
Of veterinary drugs in animal products.
Yeah, animal food product. I mean, probably animal products consumed by the human. Consumed by human.
So that would be like for any other. So that is mostly true with the meat and the meat products that are, or maybe say poultry, eggs, etc. So those levels are decided by in the various FSSAI requirements guidelines that a particular food commodity may have these levels of these components XYZ, whatever components that are being referred to. So it will be mentioned in the FSSAI guidelines.
Yeah. Okay. And I think there is no much questions. And the last question from my side also is about the opportunities in this field of regulatory toxicology for the students, veterinary pharmacology and toxicology students. So how is the opportunity in this field?
So there are a lot of opportunities as in most other fields. A regulatory toxicologist would be required, or a regulatory toxicologist would be a kind of a liaison between the regulator and the particular industry or the particular um, organization. So either in any of these food, feed, chemical, pharmaceutical, all these industries have openings for regulatory toxicology. For toxicologists per se, over a period of time, then when you further say specialize in these guidelines, regulations, etc., they would then get into the realm of regulatory toxicology. But various industries, then there are various scientific institutions, organizations say CSIR also has several toxicologists, ICMR. So both in the private and in the government sector, there are organizations, there are say companies which recruit and also require services of toxicologists?
Okay. So that's all the question. I think now let me invite Dr. Sailesh Bhavsar to present the vote of thanks. Yeah,
Uh,
sir.
yeah. Thank you, Dr. Srinivasan. I am highly thankful on behalf of the ISUPT to Dr. Akshay Dwarkanath for a nice presentation on regulatory toxicology, challenges and opportunities. And it was a nice, very informative presentation lecture and a thought-provoking ideas you have given. And there are a lot of questions have been generated which will be useful to the
Wanted to tell you to unmute
Dr.
more.
Akshay.
Unmute. Oh, okay, I got it. I got it. I'll speak.
Okay. I am also thankful to all the participants from the ISVPT as well as participants from the all other say non other than the ISVPT, those students and other participants from the academia and industry, those who have participated uh, in the webinar very actively, especially Dr. Nitin Bhatia for providing a good insight into the veterinary products toxicity. So thank you very much, Dr. Nitin Bhatia also. And I'm thankful to all the executive of the ISVBT, all the senior pharmacologists for remaining present during this webinar, making this webinar a grand success. And I'm also thankful to our president, Dr. A.M. Thakur, for making or allowing us to conduct the webinar. And at last, I am thankful to Dr. Srinivasan and Dr. Ratnadeep for working as a team member to making this webinar series a grand success. So once again, I thank each and everyone for remaining present and making this webinar a grand success. Now over to Dr. Ashwin Thakur sir for his final remarks. Thakur sir.
Yeah, very good afternoon, all of you. Especially today, we have our senior most life member of ISPT, Dr. Shushma Rishi, who was the professor at HAU, and she has taught me pharmacology during my PhD studies. So I welcome her, as well as Dr. Nitin Bhatia, and as usually our KK Sharda, Dr. Ramesh, Dr. Shujit, Rohini Roy, etc. It was very nice presentation, Dr. Akshay. All of us have been enlightened with the toxicology, particularly regulatory toxicology. In fact, any product which is being used for the health of either animal or the human being is to be tested, and certain guidelines are already there. No doubt, guidelines are there, but they are being not followed at certain points, and that's why that creates the problems. So definitely for the alternate therapy, there are to be developed. The toxicology points of view or the criteria are to be developed and then to be studied, because a lot of drugs of the alternative therapies are available now. During the COVID also, many of us have used even kata and this and that, and many have suffered also from certain adverse effect or the toxicity also. Therefore, some guidelines are to be developed for the alternate medicines, particularly the Unani or the homeopathy or the even herbal based drugs. Therefore, so this webinar is very much useful to all of us. Dr. Akshay, I thank you profoundly from the ISPT society point of view and
Yeah,
from
thank
all
you.
the participants.
Thank you so much.
Yeah, and I also thank all the participants, particularly the senior most also, and Dr. Nitin Bhatia, and Dr. Bhavsar, and Dr. Srinivasan, etc., for making this webinar very successful. Thank you. I hope all of you will again join whenever our webinar is there, ISPT webinar. It will be very much useful to all the participants. Thank you. Thank you all. Have a nice day.
Thank you so much, everyone.
Thank you, sir.
Yeah,
Thank you.
Dr. Srinivasan, yeah, please.
Yeah, so the feedback link is given in the chat box. So those who are interested to get the e-certificate, kindly fill the feedback form after clicking the link. So by now, I think we can close the webinar. So please permit me to close the webinar, sir.
Yeah, Dr. Srinivasan, you are permitted to close this platform, webinar platform.
Thank you, sir.
Thank you.
Thank you. Bye bye.
Thank you. Bye.